Okay, well, thank you all for enduring the last 2 days. Looks like it was a great meeting. What I'll talk about today is our approach to single molecule proteomics. I showed you this slide at the beginning, but I wanted to again go back and kind of recall the numbers here. So we're trying to, DNA sequencing is pretty mature. Still, requirements for DNA sequencing are high in terms of the input requirements, what you put in. And then if you want to sequence native DNA molecules, you still need a lot of material. There are definitely efforts to be made in order to reduce that amount as well, and to get into access of single cell contents. Same thing with RNAs, picogram level. Perhaps this is even exaggerated, the number here. And then the proteins, you have pretty high mass, but if you look at this discrepancy is a lot smaller than this because the mass of proteins is a lot smaller than the mass of the RNAs that encode for it. That makes sense. But there's still a whole lot of proteins and probably a lot of variation in within each protein type in terms of sequence modifications and so on. And what our lab does is basically develop, is a small piece of a very big field, which is aimed at developing new methods for studying single molecules. Among them, we're going to be studying proteins, and I'll highlight some of the challenges that we face in the field in terms of nanopore analysis of proteins. So when you look at, for example, a piece of DNA, DNA is a great polymer. Like it's a uniformly charged polymer, especially double-stranded DNA. It has this very kind of expanded form when it's free, when it doesn't have any proteins that condense it and make it all tied up into knots. Typically, DNA is very well behaved. Single-stranded DNA and RNA become a lot more complex. So when you want to kind of linearize them and study them through any sensor, not just a nanopore, but particularly a nanopore, then you have to kind of overcome some of these forces. And what has worked really well for DNA and for RNA is to conjugate the DNA to an enzyme, and the enzyme basically starts to unwind and move the nucleic acid through the pore. With proteins, the situation is very different. There are molecules which process proteins, they transport proteins. So there are, enzymes that will actually unfold and transport proteins across pores. But if you look at the chemical diversity along the protein, right, you have 20 amino acids and then some modifications. You have so many different types of chemistries: hydrophobic, hydrophilic, charged, uncharged, hydrogen bonding, so on. All these types of interactions lead to very complex chemistry, which gives the proteins the structure that gives it their function, their unique functions. So we're facing a problem of basically overcoming all these chemical interactions, linearizing them, passing them through a detector, and so on. All these problems are kind of need to be solved if we want to be able to kind of scan proteins using nanopores. So what I'll talk about today is just highlighting some of those challenges. The colors are not by any means indicative of us having succeeded anything or not. They're just happen to be random colors. But unfolding the protein prior to translocation, the linearization problem, right? That's something that we have to face. We have to overcome secondary structures. There's a lot of chemistry involved. And then there's some chemistries which we have to kind of break up in order to linearize a protein. The second problem is threading. And the threading problem is basically somewhat related to the first problem, but it's really, we're talking about getting the end of a molecule to get it into the pore. The protein will have two ends, right? If it's a linear molecule, there may be some other more complex structures, but the most reasonable way we think of a protein is just to have a single N terminus to C terminus linear structure. So threading that into the pore is a problem. The third problem is transport. So how do we drive this molecule through the pore in a way that we can kind of scan it and read the signals? So we need to figure out a mechanism, and then we need to think about the following question: do we need to use some sort of an enzyme analogous to DNA and RNA, or will we be able to just scan a protein and look at signals specifically enough to be able to discern different kinds of protein without an enzyme? For DNA, people have hypothesized both. for a very long time until the enzyme really won. Right? The enzyme won big time. And it's a lot slower. So the arguments against enzymes is, you have to wait for the enzyme to move. And then the other argument is the enzyme can die and can, single molecules denature and so on. And then the readout. So this is the most important part, because if you're able to do all of these things, you still need to be able to get the information that you want in order to reconstruct the sequence of the protein. So you need to have a good enough pore, and you need to have enough time to read each segment of the protein. All right, so now I'm going to jump to talk about basically a story about how we kind of meandered around solving these problems one by one. And, of course, we haven't solved everything, but we solved numerous aspects of this. So the first problem is linearizing, right? So in order for the protein to be linearized, you have to overcome a lot of these chemical interactions that are intramolecular to the protein. And one way to do that is to use something called a denaturant or chaotrope, like, for example, guanidinium chloride or urea, right? But in order to do that, one has to basically take the protein and immerse it into a high concentration of this denaturant. And it turns out that lipid membranes are not very comfortable with having high concentrations of these denaturants. They tend to be leaky, and they tend to kind of break up. So I'm talking about DPHPC or whatever membranes people usually use. So we had to, the second part of this, so this is kind of a multi-step problem, bear with me. The second part is that proteins don't have this backbone charge. And when you don't have a backbone charge, driving the protein through the pore requires you to have sometimes a higher voltage than typically that you apply for DNA, because the voltage is the driving force of the molecule through the pore. That leads to various modes, including electrophoretic forces and electroosmotic forces that capture and drag the molecule through the pore. So we had to develop first a system that allows one to apply a reasonably high voltage, and then we had to develop a polymer, which is a substitute for a typical lipid membrane that actually overcomes the issues of the chemical sensitivity to denaturing agents. So this is the first part of the problem, where we basically designed an aperture which will be a support. This is not where the pore is. This looks like a pore, but it's 100 microns. It's way too big. It's about 10,000 times the size of a protein molecule, but it does support a planar lipid membrane in a way that allows us to then paint lipid, literally paint lipid across this aperture, and then insert a protein molecule, which serves as the pore, as the sensor. So basically, we generate this planar lipid membrane. This aperture, it's a little bit maybe difficult to see from your angle, but this aperture is here. It's very planar, and it has a very thin wedge structure, which leads to very high stability of the bilayer. That's actually a critical thing that has to be solved if you want to apply high voltage. And then we tested the insertion, and we tested this at high voltage, that we were able to apply a large voltage, even for 100 micron apertures, which are very large. The size of the aperture here is important, because the larger the aperture, the easier it is to insert the protein. Smaller apertures tend to take longer if your protein is not very active. And we want to test different kinds of proteins. Some of them are active, some of them are not. And when I say proteins, I mean the pores, not the proteins we're going to study. So we made this aperture, and then now that we have this aperture, the second part was adding the denaturant. And we chose guanidinium chloride. The reason was that we could have chosen urea. Those are the two main ones that people use to denature proteins. Guanidinium is a charged molecule. Urea is neutral. And when you add urea to it's also viscous. It has viscosity. It increases the viscosity of your solution. So when you add urea at high concentrations that are high enough to denature proteins, it actually increases viscosity, which reduces your ion current signal, because the mobility of ions is proportional or inversely proportional to the viscosity of the solvent. So you lose signal. And when you lose signal, you lose resolution, because the number of current levels you can detect depend on how much current you have going through the pore. So we naively thought that guanidinium chloride would work better because it's charged, so it would help us get more current, and it does. However, it causes a tremendous amount of leakage through normal bilayers, and we had to seek alternatives. And one alternative that my graduate student, who's basically graduating in a couple of months, came up with is this polybutadiene polyethylene oxide. It's a very nice commercially available material. You just buy it and you basically dissolve it in chloroform, and then you can make your membranes, and they're very stable up to, say, 350 millivolts, which is a pretty high voltage. You can use these things at very high concentrations of guanidinium chloride. We used 5 molar in this case. So that was, we're basically breaking down the barriers one by one. Now that we have this support, we can then insert a protein pore. In this case, we used alpha hemolysin. It's a very kind of well-established pore, very, very stable. And we wanted to study, I'm going to skip that slide. That just shows stability. We want to study basically how this pore can be used to unfold and translocate proteins. This slide is important because it shows us how, in the presence of very high guanidinium concentrations, one can argue that the pore itself, which is the protein that is doing the sensing, can actually degrade, can actually denature, right? Because it is a proteinaceous pore. It turns out that it takes a lot more than a few molars of guanidinium chloride to actually disrupt the network of this assembly of proteins that form the nanopores. So hemolysin is pretty stable. At 2 molar guanidinium, not only does the pore component remain stable, but also this external part. So this part resides in the membrane, this bottom part. This is where the hydrophobic residues that insert into the membrane are. And then the top part actually stays aqueous. It stays on above the bilayer. And that part has been hypothesized to get to disrupt, to basically chemically denature in the presence of very high urea and guanidinium concentrations. But we've basically disproven that because we see here, we have these probes, which are basically blunt hairpins of DNA that we can use to assess the internal structure of this vestibule. This is based on work that was published 20 years ago by Mark Akeson's group, where basically these hairpins provide us with a very distinct current level. You can see here, 6 base pairs is about 40 to 45% amplitude of blockade, and then 4 base pairs is 35. So when we add the 2 molars of guanidinium, which denatures most proteins that we're going to study, it still remains stable. You can see here the dwell times get a lot shorter because the DNA hairpins themselves, the base pairing actually gets disrupted, but the overall structure of the vestibule is conserved based on our measurements. And just anecdotally, you see here that every event that we see from every one of these hairpins results in a very deep dip, and that is actually the hairpin kind of melting and going into the pore. When it goes into the pore, it produces a deeper blockade because now the DNA will reside in the tightest constriction, and the tightest constriction produces the deepest spike because it blocks most of the current. Because ions can just go around this hairpin when it's here, but once it gets into this, the DNA gets into the tightest constriction, most of the ions are blocked. And that's what you're going to see with proteins as well. So we wanted to basically then test our hypothesis that proteins can be linearized and threaded through the pore. And we constructed the system again and added proteins. But in order for threading to initiate, we had to use a charged tail. The charged tail is essential because you can think of the protein as this polymer that's kind of, even in the denatured state, it's sampling space. And there's no particular reason for the end to thread unless there's a charge, unless there's an electric field operating on it. And we've shown that as well. You can see that when there's no tail, basically the event structure, the sample set of events results in very just random, events of varying amplitudes and duration that do not correspond to any ordered process. But when this tail is added, which is only 10 amino acids of aspartate, then threading initiates, and I'll show you that. So we made just a few different types of proteins which vary in their content. So the sequence content, like we took GFP, which is notoriously difficult to unfold. We wanted to show that we can unfold it and pass it through the pore. We took maltose binding protein. This is the wild type. And then we added N-terminus tag and a C-terminus tag. And then we took a dimer of the same protein and added a C-terminus tag as well, because we wanted to see what is the structure of the signal going to give us if we have a dimer of the same molecule. So it should give us a longer sequence, but also it should give us that signal that reproduced multiple times. So our first experiments, again, I'm going through the story of kind of like the hard labor that my student had to go through. Our first experiments were done with 1 molar guanidinium because we saw denaturing curves in bulk, and we saw that 1 molar is pretty unfolded. And pre-unfolded at the single molecule level, unfortunately, is not unfolded enough. And what you see is that you see the top here represents the baseline current, and then you see these blockades. And our first event here that we're seeing is extremely long, and that represents a protein that's not fully unfolded. So basically, some of it gets into the pore, but then it does not have enough of a driving force to get through because there's secondary structure that's still present. And that was hard. That was only a hypothesis, so I couldn't have told you this unless we did the following experiments, which were to increase the guanidinium concentration. And what you see in the scatter plot here, you see dwell time on the x-axis, and you see current blockade on the y-axis. And you can see that there's 2 populations that I've highlighted here. One of them is very kind of tight and centered around 85% mean blockade. So 85% of the current is blocked, and the mean dwell time is a few milliseconds. And then there's another population that is slightly shallower, so less blockade, but also it stretches over time. So some of the events are faster, some are slower, and they can extend up to a second or even multiple seconds. So that larger population, or the broader population, is the thing we were trying to eliminate. And by increasing the guanidinium concentration, we noticed that they completely disappear. And you basically get, all of your events now become, in this tight space, which tells us that now we have a controlled, ordered process where every protein that gets in gets out. As I mentioned, this tail is quite important, and I wanted to show you what happens when the tail is not there. And actually, our group is working hard to make those tails post translation. So we don't want to engineer a protein to have that tail. We want to actually take native proteins and study them. So we want to terminally tag these proteins with tails. But in this case, we used the engineered version, and we saw that if you just look at a trace here and its corresponding scatter plot, all these events which correspond to interactions of this protein with the pore are basically all over the place. Mostly they're kind of shallow. And if you recall, shallow means that the protein is basically hanging around the pore, but not really making it all the way in. Whereas if you take the tail version, you see very ordered events. Most of the events look of reasonably similar depth. And if you look at the duration, which you cannot see here, you can see that here it's kind of a tight distribution. And in fact, we can model that distribution according to a model that I'll show you later, and show you that this transport is kind of ordered. There's some method in this transport process. Now, going back to the unfolded and folded population, I'm showing you something here which usually people turn their heads. I see everybody turning their heads, but I don't see it this time. That's good. Don't turn your head yet. You don't need to. I'll explain to you what I'm showing. The dwell time here is on the y-axis. I just rotated this plot. And the reason was what I wanted to show you is that there's a highly extensive set here of going all the way from low force or voltage all the way to high voltage. And you can see that at the very high voltage, this population, which corresponds to the linearized protein, it basically is the fastest. So the dwell time is the shortest. The pulse width is the shortest. And as you decrease the voltage, you can see that shift all the way down to, almost an order of magnitude slower at almost an order of magnitude lower voltage. Now, the other population is the one that corresponds to the partly unfolded. That one is much more sensitive to voltage, as expected, because that one represents overcoming secondary structure. There's a barrier. And in fact, the voltage dependence of this population is a lot stronger than it is for the kind of freely flowing population. And when you look at the velocity of the protein, which is a mean value, we don't have any way of measuring instantaneous velocity or the velocity as a function of time because we don't have markers on the proteins. We just know that the sequence is there and we're passing it. But if you take the total dwell time and you know the contour length of the protein, you can calculate what the mean velocity is. And you can see that it responds very well to voltage. And we did this for both monomer and dimer protein. These dots here represents the values at a higher guanidinium concentration, 2 molar, which we did at this voltage, which I'm going to show you most of the data from in just a bit. Now, what's interesting here is that the protein is going through the pore with a velocity that responds to voltage. We don't understand the mechanism of it. So we did, we didn't understand the mechanism of it. Now we think we do. We did molecular dynamic simulations of this process, but with the group of Alexei Aksimentiev. And what he did is basically an all-atom simulation of the pore embedded in the bilayer with and without guanidinium chloride. And it turns out that, we all know that guanidinium denatures proteins, and we all know that it happens around 0.5 molar to 2 molar, the denaturing curves. So basically, one can see in the simulations as well that guanidinium not only denatures proteins in bulk, but it also binds to our protein. So our protein itself has affinity towards guanidinium. The affinity is very weak. It takes a few hundred millimolar to get that binding to occur. But the consequence of that binding, which it's very hard to see here, but there are these pink kind of structures that are immobilized, and we can plot their position and their kind of how long do they stay bound to the protein, and they stay much longer than the ions do. And that results in something very useful for us. That results in basically converting that protein into a more positively charged wall. So the binding of guanidinium to our pore actually generates an electroosmotic flow. It generates a positively charged kind of lumen, which actually responds by having chloride being the main current transporter. So usually potassium and chloride kind of share the load. They have similar mobilities. Yeah, and they basically this chloride actually generates all of the force that's needed to drive the protein through, which was very interesting. And these are the distributions, and it doesn't matter if you have N terminus or C terminus and entering the protein enters whether this from the C or from the N terminus. We get very similar dwell times, and they're very much a function of the length of the protein. So here you can see the dwell time, and basically these. If you plot the length of the protein, like the dimer basically is twice the monomer and so on. So the velocities are very similar. And what I wanted to show you, I skipped one slide here. The N terminus versus C terminus are basically indistinguishable in terms of their bulk population analysis. If you just look at each molecule and you say, what is the dwell time and the amplitude, you don't see a difference. But we can look at the signals, and we do see the differences in the signals. This is the C terminus protein and the N terminus, and they have virtually inverted signals, which tells us that we're reading something about the protein. The dimer also has kind of a double, and these are dynamically time-warped events overlaid. So it might be a little bit difficult to explain in the short dwell time, the short time that I have. By taking these signals, we can actually then we can actually apply machine learning to classify whether this molecule is maltose binding protein, green fluorescent protein, or a dimer of maltose binding protein, and we get reasonably good accuracies. To improve those accuracies, we need higher resolution pores, and that's what we're working on now. We also did this for GFP and MVP, and we saw for different ratios we can recover. And you see here, this is I just need one more minute. This is a 20-80 ratio of maltose binding protein to green fluorescent protein, 50-50. And these are the classifications from single events. So each event we can classify which protein it is from the binary mixture. And then 80-20. And then this is for different ratios. You can see we can recover it quite well. So we can really identify, but we can identify all of the proteins. That's the thing I was saying before that in my talk yesterday is basically that you need some form of separation followed by classification. We are now making other types of pores which have enhanced electroosmotic flow by engineering the proteins, by changing the inner content of the pore, and we're able to get This is a trace from one of those mutants that worked really well. I can't talk too much about it yet, but it's going to give us a much better resolution. And what I mentioned, the last point I mentioned, which is basically on the speed. In order for us to be able to have we have to have a good pore, so it has to have a high enough resolution, and we have to have enough sampling time. To get that sampling time, we need the residence time of each amino acid in the pore to be high enough. And these pores actually provide us with 6 times better residence times than the previous pores that we've made. So we're very excited about these pores. And that's it. And that's the summary. So this is the student who's learning. Yu is the student who's about to graduate and go for a postdoc. And Shinchi developed the aperture, and Amar and Ali have done the machine learning and developed all of the analysis software. And we're very excited to move on to fingerprinting a wider set of proteins. And thank you very much for your time.
Thank you for the talk and sharing your journey with us and teaching us a lot along the way. It's nice to think of life in Reynolds land. Any questions?
Hi, great talk. Maybe a very naive question. From very high level, this looks a lot like what the cells are doing in proteasome. Have the field tried it? And if not, what's the challenge? Because you could eventually do even from protein mixtures, right? The cell knows which proteins it wants to take to the proteasome, and then it linearates and degrades.
Correct. So, absolutely. So actually, there's Jeff Nivala at University of Washington is actually very much working on the system that you mentioned. And as long as you have a tag, that SSRA tag, I believe, which helps the ClipX system recognize the protein, then it can load onto it and basically get pulled through. So that is something that is a tag that needs to be there if you want to analyze all the proteins. So that's kind of the downside. No one has to engineer that in, just like we have to engineer our charge tag. And the thing that is not present yet in any of the systems that we're dealing with is a way to control the structure of the peptide that goes through the pore, how stretched it is, like how tight that force is. You know, with DNA, it's very easy to pull on the rope when you have force, but when you don't pull on it, you get this entropic mess, right? It's basically just, you know, a random collection of, of a chain that's basically either bound to the pore, unbound to the pore, moving around, going back and forth. And controlling that really requires a uniform and large enough force. And I think that's where the challenge is, that we're, with our guanidinium system, we're able to overcome some of these challenges with unfolding and stretching. So potentially, if some of that enzyme machinery is compatible, you know, with some levels of denaturance, for example, on one side, or, you know, some configuration, that could be a good way of going about combining these things to achieve the readout. Yeah. Sure.
In terms of, you have to engineer, there are methods called targeted degradation, where you go for a protein with a small molecule, and you you basically bound E3 ligase through that, and that ligase gives the targeted protein that tag you wanted. And is it targeted protein degradation? I'm not in the field. I just know that it's a thing. Yeah. So,
well, you know a lot more than me, that's for sure.
You don't have to have the tag on the protein beforehand. It can be recruited for the proteasome out of protein mixture. It doesn't have to be.
Yeah.
So
yeah, all these are great ideas. I think at some point one has to pick what bottleneck one wants to solve. There's so many bottlenecks. Getting the right pore, getting the right kind of motion, and then once everything is done, maybe integrating and working with others. And I think it's Something we've not in the community we've started is a single molecule protein sequencing conference. 3-4 years ago, it was started, and we had a third meeting in Netherlands last year, and next year I think there'll be one as well. That's where the community is really trying to kind of learn from each other. What are the different tricks, and reaching the same bottleneck in a way.
Very exciting progress. I think that one of the important next steps will be to generate a large enough training data set so that one can associate electric currents that are measured with protein sequencing. Do we have any estimate as to how large that training data set has to be to achieve to something that is generalizable? Because with the current approach, I feel reasonably confident that if you increase the number of proteins that you analyze to maybe 50 or 100, you maybe can discriminate between all of those proteins in the data set.
Yeah.
But then how do we go to something that is more generalizable? What are your thoughts on that?
Yeah, no, this is a great question. I think at this point, I think we're this is really still in diapers. We're still trying to optimize what the best pores are and what the most general approach to linearizing and transporting proteins. There are still some issues we came across. For example, what happens when the protein is charged in the extreme direction, when it's an extremely positive protein, when most of our proteins here are negative that we study, but they're very slightly negative. Slightly positive proteins can also go through. But then if you really have a really positive protein, then it will get stuck, right? Because you have a competition between electrophoretic and electroosmotic forces. So I think we're really, that particular data set that you're referring to, I think is somewhere along the line of maybe 3 bottlenecks down the line, after we have the right pore, after we have the right mechanism for motion and the right throughput. And throughput is the key. That's where integrating many pores together. We're making like 4-channel devices now in my lab, so we can increase our bandwidth by 4. But it would be nice to have 100-channel or 1,000-channel device, and then maybe we can really get to the kind of questions that you're referring to.
Thank you.
There's one in the back there and one in the front.
Then I would just ask one in that case, and this is really exciting work. So I'm more familiar with the microscale electroosmotic flow, and then you talk about the model in the nanoscale osmotic behavior, which like I'm trying to understand how stable that that charged wall is. When you push a charged protein through it, how much interactions you're getting from that. Yeah,
No, these are great questions. Actually, modeling the electroosmotic flow at very thin walls, very kind of narrow, right? Like, do you get a plug? Do you get, what kind of flow do you get? And it's not. I would say it's there. I have to assume it's there because it's working, right? And if we tune the charge, we see that it disappears. So we can really tune that process. But what is the nanoscale, equations that govern that? That's actually there. Numerous review articles and on modeling these systems when the thickness of the capillary basically becomes 0 or close to 0. Not trivial. So this is an excellent question for perhaps theorists to tackle.
Thank you for the wonderful talk. I have a follow-up question on Nicolas' question. So in order to achieve the discrimination of proteins or classification proteins from a super complex mixture of proteins like 0, we definitely need a machine learning algorithm trained on a large data set. So my question is, do you see there would be an ideal pore, and what could be its ideal structural or chemical properties that we want in order to achieve this, or it will be a mixture of a class of pores? What would be necessary to achieve that goal?
Yeah, I mean, I think we can only. I can only extrapolate from what works today, and if you'd asked me about what works for genomic sequencing and whether nanopores would ever sequence DNA, and now it's even ludicrous to say that I didn't believe that, right? It's like, I don't know, I'm not the only one who didn't believe that it would work. in my field, but when it works, then okay, so what works about it? Let's try to model that and mimic that. So what works about it is that you have a way of pulling something very effectively, but you also have a way of controlling the motion very effectively. So you have that coupling of pulling and controlling the motion. And the third piece, which is, we didn't even talk about that, is the extent of resolution that you need. In the case of proteins, you need an enormously high resolution that is maybe unachievable because with DNA, you're reading several bases. You're never reading one base at a time, right? You're reading a k-mer of 4 to 5 different bases at any given time. When you're talking about proteins, 5 to 6, maybe 7, because the backbone chain is a little bit shorter, significantly shorter. So you might be reading 10 amino acids at a time. Just giving so think about how much different permutations you may have. However, the permutation space of proteins that we are aware of is a lot smaller than all the combinations of amino acids. So one can recover a significant fraction of known sequences. And I really think that we've reached an era where we need a lot of controls. We have machine learning, we have AI, we have a lot of tools, computational tools to tackle these problems, but we really need synthetic controls. And even for like RNA, we're making a lot of control molecules with control modifications in order to assess how quantitative is our tool, our method. So I think to answer your question, what is the best pore? I think integrating a good high resolution pore requires, very tight constriction and very kind of shallow constriction, but one that accommodates the largest modification, post-translation modification. So that could be a challenge, right? Some proteins will get stuck if they're modified with a big, bulky PTM. And then you need a good force. You need a good way of generating force, yet controlled motion. So a lot of issues there, and some of them are conflicting directly, right? But I think finding the happy medium is basically, and the thing what I wanted to mention is that you can cut the protein up into peptides, and then you could probably do things a lot faster in terms of short term with recognizing which protein you have cut. If you've separated, a class of protein like in a gel, and then you wanted to pick a piece of the gel and analyze it, you'd break it up and you would look at all the components of that. You'd probably be able to isolate tens to, if not, maybe 50 different, species, right? And then if you have control, you might be able to identify those species. But I still believe in full length protein analysis. So I'm not gonna. That's kind of like the niche where I can hopefully make a dent. Right? Other people are also digesting proteins and looking at that with nanopores. I think their mass spec it blows everyone out of the water personally. So I don't think, but full length protein, and of course people are doing that too. But full length protein analysis with nanopores could be a good way to tackle some of these.
Great. I know there's a lot more questions, and that's wonderful and really exciting that people have an interest in something that's quite different, looks at things with a different lens. But I think we need to move on for time's sake, but I think hopefully Professor Wanunu will still be here and can chat with people. Gentleman over there had a question for you too. So
yeah, love to talk to you.